I threw this together a few years ago for the Bioinformatics BSc, and at that time I must have borrowed from these guys at the University of Arizona. I have no recollection of actually compiling this talk, but there we go. So, why bother to read papers? Well, journal papers are far more current than textbooks. If you try to read a textbook, it takes years to get a textbook into print. So anything you read in the textbook is pretty much by definition a bit out of date. You should also be getting enough details and methodologies by reading papers to replicate what you read about, so you can start actually looking at what is the cutting edge in terms of approach and use those approaches in your own research. So those are two important reasons why you should bother to read papers rather than just read manuals and textbooks. It's also part of training your critical faculties. So there comes a time where you realise that just because a thing's put down in black and white somewhere doesn't mean it's true. People can put out all sorts of tosh and nonsense, both in textbooks and in papers, and actually going through the papers and reading them and wrestling with them is actually good. And this was for students, undergraduates, but obviously for you, the day when you're writing your own papers is now. And the only way to actually write good papers is to have read a lot of papers to work out how other people do it. And whenever I want to start writing a paper on a particular topic, I will go and read all the papers on that topic and look how they crafted their introductions and their discussions to sort of see the threads that they put down. So, you know, all of that's useful. So there are various kinds of papers out there, original research papers, which are the kind of, those are the ones we're supposed to be aiming for. Those are the ones, the only ones that count in the Research Excellence Framework pretty much. There'll be others out there, reviews, opinion pieces, hypothesis pieces. Sometimes those are actually quite insightful. Certainly if you're coming into a new area, reading a review is a good way to get on top of it. Sometimes people blend in actually bits of research with opinion. That's kind of deprecated, but it does happen and it's sometimes useful. The key point is, the paper you're looking at peer reviewed, or is it invitation only? Or is there some kind of shenanigans gone on where a guy has been asked by his mate to write a paper and it's had a light touch peer review? You know, a lot of stuff does get published that is not peer reviewed or only lightly peer reviewed. So the value of the paper, the weight you put on it, depends on how critically it's been peer reviewed. There's a lot of discussion actually lately about whether the peer review you get in papers in journals like Nature and Science is different from the peer review you get if you publish in a discipline-specific journal. And there is even this heretical thought that actually the peer review that goes into a Science or Nature paper may not be as good as in a discipline-specific journal. High impact journal, if it's in a high impact journal, the first approximation you think, oh, it's got to be good stuff, although again, as I say, that's been questioned. And the author's reputation, if it's from someone who already has an established reputation in the field, then I guess you're going to think this is probably okay, but even eminent people publish tosh and nonsense at times. So I guess nowadays we probably don't have to say much about citation rates and impact factors because the whole of the scientific community is obsessed with these bibliometric factors. But you can judge a paper by how many times it's been cited. And you can find that out now with Google Scholar very quickly. And you can look at journals, and to the first approximation, you can judge them by the impact factor, which is worked out as a formula as to how many times the papers on average have been cited in that journal. I think it's over a 2-year period. You need to think to yourself, is this a specialist journal or a general journal? If we're looking at bioinformatics, which, as I say, this was written for the Bioinformatics BSc, a number of bioinformatics dedicated journals in that field. So papers generally conform to this so-called IMRAD format, which is Introduction, Method, Results and Discussion. That's kind of the bog standard approach. Some journals do play around with that. Some journals take the view that nobody wants to read the methods really, so they put the methods right at the end and they have the introduction, results, discussion, and then the methods are hidden away in a different typeface. Some journals, things like Science and Nature, they actually hide a lot of the methods away from the body text anyway, so they're either in the footnotes or they're buried in a supplement, an online supplement. But you know, most journals have something like methods, introduction, methods, results, and discussion. Of course, there's other things in there. There's the title, the abstract, authors, acknowledgements, declarations, references, tables and figures, and legends. There is a great pressure on the length of a paper when it's printed in a printed version of the journal. So many journals will say, you know, it's 6 pages or whatever, because the traditional idea was it was going out in print and it cost so much money and so forth. That some of that is disappearing with sort of the open access journals that are only appearing online. They're happy to take papers of any length. But if you're trying to get into PNAS or if you're trying to get into Science or Nature, then there is a pressure on the length of the thing. And there's also this issue about making it accessible to the non-expert. So if you're trying to get into Science or Nature, you have to write your paper for a general audience. You're not writing it for another microbiologist. You're writing it for the average, you know, you're writing it for Professor John Heath, who is not an expert on bacteria, but might be persuaded to be interested for 10 or 15 minutes in his busy life in your paper, if you write the paper in an accessible way. Some papers combine the results and discussion, and you could, again, there's kind of argument, discussion about whether that's a good thing or a bad thing. One view is that you should keep them apart. So the results are the results. That's what you found, and your interpretation of them and how they fit into the bigger picture is a separate thing that should be put into a separate section. That would be one view that some people would take. And you shouldn't muddy the waters by mixing the two together, because then the reader can't tell where do observations end and interpretation begin. The counter argument to that is that sometimes you have a narrative that runs through. You do a load of experiments on this thing and you get some results. On the basis of those results, you then decide to go and do another load of experiments, and you get some conclusions from those, and then you go and do another experiment. So if you try and write that up, fully compartmentalize into results and then discussion and methods separate, it starts to become a bit difficult. So some journals will allow you to have that kind of narrative style. I've mentioned most of those other points there. So the first thing when you approach paper, it's not a novel. It's not like reading Anna Karenina or War and Peace. You don't have to start page 1 and read every page in linear order until you get to page 500. There's no requirement to do that at all. You don't need to go into that linear approach. So what you can do is you can quickly skim through the paper and look at the title, see if it interests you. Look at the abstract, which tells you what's in the paper, what's interesting. After you look at the abstract, flick through and have a look at some of the figures and the tables, because those often contain the meat of the paper, the actual stuff that people are trying to convey. And then, once you've done all that, you might, if you decided after doing that that it's worth continuing and this really is an interesting paper for you, then you look at the introduction, the results, the discussion, and most people would only look at the methods after all that.
There's that interesting psychophysics study of scientists reading papers.
Oh yeah.
Where they, give up some piece of electronics that apparently most people scan text looking for keywords.
Yeah.
And they will read around the keywords.
Yeah.
They don't read from the beginning of the paper.
Yeah, exactly. Yeah.
The name of their favourite gene.
Yeah, yeah, exactly. You just skim through it and you get, yeah. In a way, reading a paper it's not just reading a paper, it's a struggle with the paper. It's kind of a wrestling match. If you just sit there and it's not like you're putting on a cine film and allowing it to sort of play on the back of your eyeball. You should be actually engaged with that paper. And in some ways, you shouldn't be reading a paper without a pen or a highlighter in your hand, so that you can actually then scribble on things and say, don't understand this, or this seems like rubbish, or wow, what a wonderful technique. If you do that, if you're engaged in this kind of active reading, you're much more likely to retain the contents of the paper and make sense of it. The other thing is, as I say, we don't read a paper from beginning to end and in a linear fashion. You can skim through the paper, get the drift of it to start with, and then think, actually, I don't quite understand how they've done that. But you can continue reading to the end and get to the first pass through the paper. Then go back and say, right, I really do need to understand how they did that little bit there. How did they analyse that thing? Or how did they actually set that experiment up? And you can then spiral in on those points and get to grips with them. So it should be a struggle, not just a passive thing. And you need to get into this kind of, like using your razor-sharp intellect to actually kind of joust with the paper. Get into question-asking mode. So you doubt everything. How on earth do they know everything? Everything in the paper that is said should have two sources. One is the experiments that they have done and they're reporting in the paper. The other is the scientific literature, and they should provide a citation for every declarative point they make. So the whole paper should rest on that kind of that bed of knowledge, if you like, and you can go through it and you can doubt it. So if they say it's commonly accepted that using V1 to V3 is a good way of analysing 16S data, you can say, well, I don't agree with that. Where's the evidence for that? And often there won't be any evidence. They just sort of, with a wave of the hand, they try to just go over a point. You can nitpick, find fault. Just because it's published doesn't mean it's right. And you're doing effectively peer review while you read it, making your own opinion about the paper. And the Arsenic Life paper is the one at the moment everyone's talking about. That was a paper that was complete rubbish. It shouldn't have got through peer review, but because when people read it, they immediately said this is rubbish. They were engaging their brains. They were not taking it as read, as just this is true because it's in science. It must be true, and there was an explosion on the blogosphere straight afterwards. It's never been retracted, and that's a reflection of the conservative nature of traditional print media and some parts of the scientific community. If you really want to get to grips with the paper, you shouldn't just be reading it in your own room or on the train or whatever. You should be talking to other people about it and saying, have you seen this paper in PNAS this week, or seen this paper in Science? And getting to grips with it. So, you should be saying to Jackie, have you seen this paper on the chicken resistome? And she'll say, no, I haven't seen it. And then she'll say, is it any good? And you have a discussion. And again, it makes it much more likely you remember things, you're much more likely you understand it. In many journals, there are commentaries on papers. So they're kind of like a hand-holding idiot's guide to the paper. So if the paper is a bit arcane and it's hard to read, if you go to the news and views section in Nature, it will tell you why is that paper so important, and it will hold your hand in a much more accessible way. So sometimes you read those and then you go back and read the paper and it starts to make more sense. First time round the paper seemed a bit arcane. You should be, again, as you're struggling with the paper, there may be words in there that you don't understand, and you might have to look at dictionaries or textbooks, online links to references, figure legends. You've got to struggle with it to get to grips. You can't expect to just understand everything the first time you read it. So that's all important. And just because it's in black and white doesn't mean that it's perfect. Sometimes you read the paper and you think, I can't make sense of this. And you think to yourself, am I stupid? Then you realise, no, they've just written the paper badly. Sometimes they don't explain why they've done a thing. They just plop it down on the page and they don't give you any rationale for it. So when you're reading a paper, you should look for that. When you're writing a paper, you may have to provide that in your narrative to say why you did it. Sometimes if you're too close to a subject, it seems obvious that that's what you do, but you have to explain it for someone else. Another problem, jargon and acronyms. This is a particular infestation of biomedical literature, and in fact, it's quite common just in looking at people trying to write grant proposals. They want to just put in loads of acronyms all the time. You sort of think, well, it doesn't actually. You don't save much space by using an acronym, and it actually makes it less accessible to someone who's not familiar with all your acronyms. Sometimes it can be confusing because all the side issues are given equal airtime with the main thread, and you can't quite see what they're doing. Ambiguous or sketchy descriptions of what's in there. And often someone will say that they've, we did such and such. We used a Lamely et al., let's say, for Western blotting or something, or polyacrylamide electrolysis.
But the most interesting thing is
And the author doesn't know what they've done themselves, you mean? Well, they don't understand their own experiments, you mean?
well, the data has some element that hasn't attracted the attention of the author.
Yeah. So sometimes, so for sequences, you might think, hang on, I didn't, you go look at their sequence and you find it has homologs that they didn't recognise or stuff like that. I've seen that happen before.
And also the dog that doesn't bark, which is an interesting one about why didn't they do that experiment? And then it turns out there's a reason why the experiment wasn't done.
Or it was done, but it just didn't work. And then,
so you've got to sort of look at
Okay. So when you're looking at the paper, what are the questions the paper's trying to address? What are the conclusions they've come to? What's the evidence that supports those conclusions? Do the data actually support the conclusions? What's the quality of the evidence? And actually, does the paper matter? Why are the conclusions important? Does it make any difference to the world? All of those questions should go through your mind. So when you're looking at the questions, the kinds of things that you can be looking at in a paper, you can have descriptive research, where people are just sequencing things and just describing them. There's not much understanding there. It's just a collection of, it's like mapping a new continent. Lots of facts, may not be any patterns in the facts. He's already had this lecture years ago, that man. Comparative research, where you're asking whether something is a very specific thing or whether it's actually a general phenomenon. So, for example, when I was a bit younger, I used to go and look at secretion systems and see whether they were general, found in lots of different bacteria, whether they were only found in that particular class of bacteria. And often you find unexpected things. You realise that things are much more common and general than you thought they were, and you can generalise from one set of experiments across a whole range of different organisms, let's say. Is it analytical or hypothesis-driven research, testing a hypothesis? For its own bioinformatics, you might say, can you look at amino acid composition of proteins in an organism and use that to predict thermophily? In some ways, this is what everyone favours as what science is supposed to be about, according to Popper and all that, it's testing hypotheses. But it isn't the only kind of paper that's out there. Another one is methodological research, kind of stuff that Nick is trying to write up at the moment for Nature Biotechnology, trying to find better ways of doing things, describing resources. Of course, these are not absolute distinctions. Some papers have a bit of hypothesis testing, a bit of data collection, a bit of methodology all mixed in together, but it can be quite confusing when they do that. So look at the title, abstract and discussion to try and see what the conclusions are. Do they matter? Are they of general relevance, broad in scope? How detailed are they? Are they kind of things that are accessible to a general audience? Do they speak to the whole of the scientific community? A new reason why the dinosaurs went extinct would be of interest to everyone, whereas finding that one particular amino acid is responsible for an interaction between two rather obscure proteins, well, that might not be of general interest. But it might still be important for people in that particular discipline. So look at the evidence, results, relevant tables, figures. Obviously, a paper, the conclusions are better founded if there are several different experiments or approaches that combine, kind of form an internal cohesion, if you like, in supporting a particular conclusion. Another way in which papers are written is that you'll have a first experiment that has several possible interpretations, and then the later ones are there to distinguish them, to test them, and so on. So in an ideal case, your discussion is going to bring with start with a section that sort of says, there are 3 lines of evidence for this, and then enumerate those lines of evidence and look at them critically, cast doubt on them. If all they say is that we've discovered this and that's it, then you have to treat the paper with a bit more circumspection. They should be their own worst critic as well. They should have seen the flaws in their own work. You need to understand, if you're going to judge the quality of the evidence, so you need to understand what they've done. You need to consult textbooks or other papers, looking at the limits of the methods. So, for example, in bioinformatics, some people, they slip into this idea of thinking that homology is a fact. Well, homology is a fact, but the interpretation of homology is meaning that these things have the same function, or the same structure even, is actually a hypothesis that's sort of layered on top of the homology. And even then, with distant homology, where you're seeing kind of very scanty similarity, you don't even know if it really is homology. So you have to treat that as a working hypothesis rather than say, I know that that's homologous to this. And so that's quite important. Separating fact from interpretation, I said that before. Are the results expected? And this is where this claim, I think it comes from Carl Sagan, extraordinary claims require extraordinary evidence. This is where the arsenic life thing should never have got through, because it was such an extraordinary claim that how on earth did they get there? Get that paper out. Looking at the details, assess them for plausibility. The veracity of the whole of the paper depends on the veracity of its parts to a large degree. A few years ago, I went into a PhD viva, and this guy had said that he had analysed, I don't know what it is, 100 genomes to look for the evidence of how much horizontal gene transfer there was in bacterial genomes. And I said, well, let's have a look at what you say about E. coli. Because if what you say about E. coli is wrong, because that's one of your 100 genomes, then I can quite credibly doubt everything else you've said. And he said, oh, I didn't look at individual genomes like that. You sort of think, well, hang on, if that small part of it's not right, then how do we know that when you build the whole house of cards that it actually it holds together? What are the controls? What's the gold standard? In bioinformatics, people annotate genes, and they annotate genes, make assignments, call a domain by a certain name, say a domain is a certain domain. Well, where is the evidence? The gold standard has got to be an experiment that shows you that that protein has that activity, and often that's lost in these annotation trails that are very murky. There's obviously this logical connection as well. The data may actually say something quite credible that you sort of think is okay, but then they may be overinterpreted by the authors, and they may be forcing it into a conclusion that doesn't actually hold. And there may be other interpretations, so you have to look carefully at that. So I kind of made this point earlier. If there are multiple approaches, multiple lines of evidence, different directions, all supporting the conclusions, then they become a lot more credible. The other thing that you have to look out for is hidden assumptions. Authors will often make assumptions that are hidden, and you need to look at those and say, well, actually this only holds true if that assumption is correct. So that was basically it. That's all I had in this talk. In a sense, when you read a paper, you should be peer reviewing the paper rather than just reading it. You should be trying to grapple with it and say, I'm not accepting that this person is an expert. I've got a brain just the same as them. I'm going to go through this paper. A lot of the time, if you don't understand it, it may be because you just have to work harder. But some of the time, the emperor really does have no clothes on. And when you read the paper and you think, that's not right, it's because it isn't right. And it's important to keep that in your head.